Okay, so the title of today's talk will be on novel therapeutics targeting metabolic syndrome. So by now you have a good idea of metabolic syndrome, you have a good idea of uh, diabetes, you have a good idea of lipid metabolism, carbohydrate metabolism. So it will be easier for you to understand what I'll be talking about today. So today I'll try to cover three major exciting fields in the field of in the area of metabolic syndrome, and these are some of the very hot areas in molecular metabolism. And scientists are now working on understanding these processes and trying to develop new drugs to activate these processes and help us to cope up with the metabolic syndrome which humans are facing in this century. So first, I want to talk about this uh, cell known as brown fat. So you must all have been listening about this brown fat, because we all know that when we are born, we have brown fat in our body, which keeps us warm. So what is this brown fat? Because yesterday I was talking about these fat cells, but generally I was talking about the white adipose cells, which store a lot of lipid. And because we call them white, so they are why they're called white and why this is called brown. So first of all, there are three major differences between white and brown fat. Both are adipocytes, but white fats have very large lipid droplets. So they are generally unilocular lipids. They're huge lipid droplets covering almost 90% of the cell cytoplasm. And the number of mitochondria are quite low in white adipose tissue, because it's not a metabolically active cell. Its main purpose was to store lipids and not increase beta oxidation or provide energy. Okay, so that's why they have a very low number of mitochondria, and its purpose is to store energy. In contrast to this, brown fat are brown because they have a lot of mitochondria. So the mitochondrial content gives it a brown tinge. Okay, and the lipids, they also, because they are fat cells, they also store lipid, but instead of a one huge unilocular lipid droplet, they have a multilocular small lipid droplet interspread in the cytosol, and their main purpose is to dissipate energy and produce heat. So this is a classical difference between a white and brown fat cells. Now, why brown fat all of a sudden in the field of metabolic diseases it has become so hot nowadays? The reason is that before 2007, everybody thought that brown fat is only present in newborns, in humans. And as we grow, we lose them, and by adulthood, we don't have any brown fat left. This was conception before 2007. But then something very extraordinary happened. It was more of a serendipity that this finding was made. And actually, it was in a clinic where people were actually doing radiological detection of metastatic tumors. So, tumors take a lot of glucose. Cancer cells, they just suck in all the glucose available to them. And by this, that helps in their growth. So to detect a tumor inside a body of a human being, there are ways you can tag a glucose molecule with a radionucleoside, which is called FDG. This is fluorine 18 fluorodeoxyglucose nucleoside. So what it does is that it emits positron, and which could be detected using these PET scanning. Okay, so FDG positron emission tomography is a very well-known feature of detecting tumors in the body. So these were radiologists trying to detect glucose uptake in a patient which they suspect may have cancer in the body. So strangely, when they did the PET scanning with this FDG glucose, they found that there was a lot of glucose uptake in near the shoulder region of the patients. Now, they knew that the cancer won't be here, so they thought that maybe just that some muscles are taking up a lot of glucose, and that's why this is glowing up But then they realized that this glow up of the glucose uptake was localized to areas which are meant to have fat cells. So because they was radiologists that didn't understood that it could be a discovery of a brown fat. It was shown that there are some fat cells which take up glucose, and so this is how this glucose, FDG glucose, work. So it is taken up by all the tissues which rely a lot on glucose uptake. So it will be taken up by brain. If you have a tumor cell, that will take up, so that will glow, and then any cells which is metabolically active will take up glucose. Now we know that brown fats are very metabolically active cells in the body. In rodent studies, we know that they produce heat. So what they do is they take up a lot of fatty acids and glucose and break them down and produce heat. So this is the reason why these fat cells were taking up glucose. But by that time, it was not well understood whether these may be discovery of brown fats in adult humans. Now, one of the reasons why prior studies couldn't find these marks on the shoulder of the patients taking FDG PET scan was interestingly that these patients in the clinic which were going on this PET scan were asked to sit in a room which was quite cold. So generally when the PET scan is done, it is done in a very warm environment, so there's a ease when you have this FDG injected in you. So of the many prior studies were not detecting these type of non-specific uptake of glucose in the shoulder region and the neck region. But strangely, in this case, when the study was done, it was the patients who were sitting for a long time in the cold. So the air conditioner was working in a very cold thermostat, so it was quite cold, and that is why in this study they found a lot of glucose uptake near the shoulder region. So finally, when these findings came on coming by and being reported by several other scientists, so this was all reviewed by Barbara Cannon in 2007. She published this review in which they actually put forth this hypothesis that possibly what we are seeing in a PET scan is actually existence of a brown fat in an adult human being. So this was a great discovery because now people had discovered a tissue which they thought never exists after we are growing up. So it's only in the in the newborns. So why it was important? So after 2007, the things really changed, and what people now were saying is that in man, brown adipose tissue is found in newborns and in certain adults. So the role of brown fat in newborn is to keep us warm, and in the rodents, rodents are a little different from humans. So rodents, they have brown fat always throughout their life. So the main reason they have brown fat is that brown fat helps them during the hibernation period. So even bears and smaller rodents, they have a lot of brown fat, and that could be used to generate energy when they are hibernating. So how the brown fat is induced, and why, I'll tell you why its discovery was such a groundbreaking thing in the field of metabolic studies. So brown fat, why it generate heat? So whenever you are whenever you feel cold or you have a lot of fatty food intake, your hypothalamus in the brain sends these changes, and through the sympathetic nerve system, it tells the brown adipocyte through a chemical messenger known as norepinephrine. Now, if you remember the hormones, so this belongs to the amine class of hormones. So epinephrine, and this is a very close to epinephrine, is called norepinephrine. So they are the same class of hormones, and they bind to G-coupled protein receptors. So when there is a cold sense of cold in the hypothalamus, it tells your sympathetic nerve system to secrete a hormone known as norepinephrine. It acts on the G-coupled receptor, which is a beta-3 adrenergic receptor, and activates the protein kinase A pathway. So if you remember the lipolysis pathway we studied in liver and the white adipose tissue, it is very similar to that pathway. So the triglycerides are degraded in response to this. Free fatty acids are transported into the mitochondria where beta oxidation happens. Now the difference between the things which happen after this from liver and in brown fat is that in liver there is a good efficiency of electron transport chain to produce ATP. So for each electron going through the complexes, you then you use the proton gradient and help the ATPase to synthesize ATP. But in brown fat, there's a thermodynamic break there. So it's like a it's you can say it's like a fuse type of thing. So there's the machinery is not efficient enough to produce ATP. So there's a leakiness in the electron transport chain. And this is caused by a protein known as UCP1. It's called uncoupling protein 1. So this is specifically expressed in brown adipocytes. So what it does is that it causes the protons to leak. And when there is a leakage of protons, the efficiency of electron transport chain diminishes. So the loss of energy, which is not being converted into ATP, is dissipated in form of heat. That's why the word thermogenesis is linked to brown fat activity. So you have an imperfect machinery of electron transport chain in a brown fat cells, and you release heat as a result of this difference between the net amount of electron going to the system and the total ATP produced. Now, what happens if you remove this UCP1 protein? So what it does that you can no longer dissipate heat and you can no longer cause thermogenesis. Okay, and especially thermogenesis induced by cold or norepinephrine induced thermogenesis. Now this slide shows you the effect of UCP1 or brown fat on thermogenesis. So there are two types of heat production mechanism in our body. So whenever we feel cold, we start shivering. So that is a way in which we produce heat in our body, and that is done by our muscles. Okay, so muscles are the first organs to produce heat. So there's a shivering, this is called shivering thermogenesis, and the one heat produced by brown fat is called non-shivering thermogenesis. Okay, so this is a wild type mice, and what you're seeing here is that at 30 degrees centigrade there is no shivering during at 30 degrees centigrade. This is a normal temperature, but then when you put a mice at 4 degrees, there is a lot of shivering at 4 degrees to produce heat. And if you put the animal long in at 4 degrees, this shivering goes off. So shivering goes off. So muscles are no longer required to produce heat because this work is being taken over by brown fat at cold temperature. So your shivering induced thermogenesis is reduced and your non-shivering thermogenesis is increased by your brown fat. But what happens when you knock out this UCP1 protein in a mice? So what you do is that at this is at 30 degree and 4 degree initially. There's a shivering thermogenesis, your muscles are acting, but later also after acclimatization, if you keep the mice long, this UCP1 mice keep on using their muscle to produce heat because they no longer can use their brown adipose tissue to produce. That's why this experiment tells you the role of UCP1 in heat dissipation of brown fat. So if you don't have UCP1, there is no use of brown fat actually. Now, how with humans? Do we have classical non-shivering thermogenesis? So okay, fine, we detect through PET scan that we have fat depots in our body, but are they really brown fats? Do they really produce heat? So that was the question. So to produce heat, they should have UCP1 because I showed you that UCP1 is the main protein which gives brown fat the identity, the power to produce heat. Now, there were studies done in human cadavers which showed that UCP1 is actually present in those areas where PET scan was positive. So these fat cells were not just any fat cells which were taking glucose, but they were fat cells which express UCP1. So indeed, there seems to be a functional brown fat in adult human. But can we adapt to cold? So the thing is that whenever we talk about brown fat, we talk about them only in terms of cold adaptation. So is the brown fat in human detected through PET scan is also an adaptive organ. So it can adapt to cold and increase thermogenesis. So this is again a PET scan photo which shows you that in summers a PET scan of this person and in winter. So you can see in winter there is an increased glucose uptake in the shoulder area where these bad cells or brown adipose tissues are localized. And when you see the FDG uptake glucose in summer and winters, there's a huge increase in FDG uptake in that during winter time. So it shows that human brown fat can be recruited and activated in humans just like what we see in rodents. Okay, so they are functional brown fats. Now, why discovery of brown fat was so much groundbreaking discovery? So I talked about this before. So now I'll come to that, why it is important, why it is important for us to have discovered brown fat in adult humans. The reason is that brown fat are the key to burn fat in our body. So we know that at present how we can when we eat a high calorie diet, it's stored in the white adipose tissue. The only way to burn is to do exercise, to use a muscle to take up glucose, or a liver to do beta oxidation, try to degrade fat. But what if we have another organ which is primarily only doing the thing which is burning fat? So can we activate this organ so that we can reduce the fat content in our body? And at the same time, because they take a lot of glucose, that's the reason they were detected, can we use them, can we harness their property of glucose uptake to reduce hyperglycemia in diabetic patients? So this is something like the holy grail of molecular metabolism. Can we achieve this by using or manipulating the human brown fats? So the initial studies done in animals were done in mice of with UCP1 knockout brown fat. And what you can see here in the panel A, in the control diet, you see this UCP1 knockout mice is in the white circle, and the black circle is the wild type mice which has UCP1 expression. And you see in control diet itself, if you don't have brown fat, these mice start gaining weight over time. And it is even more aggravated when you feed them high fat diet. So if you have a very high fat diet feeding to these mice, their weight even increase even more when they don't have UCP1. So and this was all done at thermoneutrality. So they were not in 4 degree, they were in normal temperature in a vivarium. And you can see that brown fat even under basal normal temperature is also very important to reduce or control your body weight. Okay. At the same time, studies also showed that triglyceride clearance in the blood is also hugely affected by your brown fat activity in rodents. So all these experiments initially were done in rodents, and this is an experiment in which they show that this is a knockout mice with a defect in the gene known as ApoA5. Now ApoA5 is an important protein regulating your serum triglyceride levels. So when you don't have these proteins, then you have hypertriglyceridemia in your serum. So there's a lot of lipid in your serum, and that may be a cause for cardiovascular complication. So in a mice lacking this gene, if you see this ependroph in which the serum is collected, you can see it is quite turbid. Now this turbidity is because of lipid content in the blood. So they are hypertriglyceremic animals. But if you keep this knockout mice, ApoA5 knockout mice, in cold, so cold is a inducer for brown fat activity. So what brown fat has done is that it has actually got rid of most of the fat in your blood. So it shows that how powerful is the activity of brown fat to reduce your serum triglyceride levels also. So it's not only your body weight, but in animals even serum triglyceride levels were reduced. And finally, what results do we have on glucose disposal? Because I told you, because glucose uptake, there is a huge uptake of glucose by brown fat, and they convert it into heat. So can we use them to reduce your serum glucose level? So in animals, it shows what we expect. You can see that these are this is the oxygen consumption rate of a brown fat cell. So oxygen consumption rate actually tells you how fast the mitochondria is working, the respiration is going on. So and it basically reflects the thermogenic potential in brown fat. So when you add norepinephrine here, there's a huge induction of brown fat activity, respiration. At the same time, there's a corroborated increase in glucose uptake also induced by norepinephrine. So glucose is being taken up by brown fat cells, which is in parallel with their mitochondrial activity. So it shows that glucose possibly is being taken up as a substrate for oxidation in the mitochondria. Now this again, this slide shows you that glucose uptake, relative glucose uptake in response to norepinephrine in different tissues. So you can see no other tissues is even close to the amount of glucose which could be taken up and utilized by brown fat when you're inducing it with norepinephrine. And this is a classic glucose tolerance test, which is generally done to see the glucose disposal in our body. So what you can see in this graph is that this is the wild type. The black boxes are the wild type mice, and white boxes are the UCP1 knockout mice. So even at the basal state, you can see the blood glucose level was quite high. So in this test, what we do is that at 0 point, we inject glucose into the animal and see how fast the body tries to cope up with glycemic surge. So this is a wild type mice, which is already has lower glucose level than a knockout mice. And then when the glucose is injected, it goes up, and then the body tries to compensate for it, and it is slowly brought down to the almost to the basal level. However, in the knockout mice, this is hugely induced, and it takes some time for the animal to bring it down. So there's definitely a glucose intolerance in a UCP1 knockout mice. So it shows how brown fat are very important for glucose regulation in our body. So these studies in the mice showed three things very clearly. First of all, is brown adipose tissue can affect obesity in animals. It can affect hypertriglyceridemia, and it can also control hyperglycemia in animals. So these are some things which are under study to see whether this can be also manipulated in humans, and the studies are going on. Now I want to talk about another variant of this brown fat. So this was discovered much later, and this is something which is even more fascinating than classical brown fat itself. The reason is that unlike brown fat, which is although present in human, but still is not a huge amount in terms of mass, but these beige fat, so why they are called beige? Because they are grayish. Okay, so they are not white, they are not brown, but they are something in the middle. The reason is that these are actually white adipose tissue which are plastic in nature. So they have a cellular plasticity. They can convert into brown like cells. So they can increase the number of mitochondria, and they can start expressing UCP1. So apart from your classical brown adipocytes, you now have a army of fat cells which can at any given time upon stimuli can convert from a white to a brown type, and these are therefore called beige adipocytes. So there are locations in mice. You have these are the classical brown adipocyte in the mice, but then you have in some adipocyte depots white adipocyte depots which also have intermittent beige adipocyte roaming there. So, and these white adipocytes, so these are very specific class of white adipocytes which can be instructed to change into brown adipocyte. This could be under cold stress or by adrenergic stimulus. Now this is how a brown adipocyte look. This is how a beige fat cells look. So this is a beige adipocyte, and you can see here that there's a it is like grayish tinge to it, and it has lot more mitochondria compared to white adipocyte, even though less than a classical brown, but it can still, and it does express UCP1. Okay, so it can do the same thing which a brown adipocyte does. but definitely you have more beige in the body than the classical brown in humans. So these are places where beige, white and brown all can be found at the same place, and these are subcutaneous white adipocyte which can be converted into beige upon cold stimuli, and these are the classical brown adipocyte, and these are the pure beige adipocytes also in humans. Okay, so what are the inducers of white to beige fat transformation? So because we are limited in the amount of brown fat we have, we cannot increase it, we can only increase the activity. So now the studies are being carried forward to see how we can convert these white adipocyte into brown, because there are twofold advantage in this. First of all, you won't have a lot of white adipocytes to store lipid, so you will lose weight in that way. Plus, you will have them converted into a metabolically active fat cell, which is which is very similar to brown, and so you can even burn more fat and regulate glucose metabolism. So that is why beige fat are even more interesting to work upon than classical brown adipocytes. So what are the stimuli for brown for beiging? So conversion of white into brown. This could be cold stimuli through CNS, and this is very similar to what we see with brown fat. So they bind to the adrenergic receptor. Then diet and gut microbiome alteration is also thought to affect the brown fat activity, and actually some bile acids affected by gut microbiomes are thought to activate this beiging process or transformation from white to brown. Then physical exercise is another stimuli which has been shown in animals to increase the production of this hormone FGF21, which act on the white adipocytes to convert it into brown-like phenotype. But the main focus is now to develop drugs or agonist drugs which mimic adrenergic receptor signaling and can be used to activate the transformation of white to brown. So this is something which is hot in the pharmaceutical industries. Can we design drugs to activate this transformation into white to brown? And recently, 6 months back, there's a groundbreaking research published in Cell Metabolism, where they have found that unlike mouse bat, the human bats express a different type of adrenergic receptor, which is called beta 2 adrenergic receptor. So mouse bat cells express beta 3, but human express beta 2. And they also said there are similar chances that the white cells which can transform into beige also express this receptor. And now they are developing specific agonists to activate beta 2 adrenergic receptor to increase both classical brown fat activity and also increase the population of beige fats in our body. Okay, so the conclusion from this is that brown beige adipocyte tissues may protect against obesity, hypertriglyceridemia, and hyperglycemia in humans. But the clinical trials will be carried forward and to see whether these things hold. So first, the drugs will be discovered, and still it's going on to make specific agonists for these receptors, and then the clinical trials on diabetic and obese people will be done. So it's quite a happening area. Now, the other happening area in this field is circadian rhythms and chrononutrition. Now, chrononutrition, as the name suggests, it relates to the day and night cycle. So it's the time cycle. So why this circadian rhythm and chrononutrition is important, because our body is always either in the activity phase or the rest phase. So we have been evolved during evolution to work during daytime and sleep in the night. That's how our ancestors were doing before the discovery of electricity. But electricity has changed everything. So now, because you have a refrigerator in your house, and you don't sleep, you can eat anytime. So that has all changed our metabolic functioning, and that is one of the basic reason of increase in metabolic syndrome in the industrialized world and post-industrialization in the 21st century. So the reason why this cycle, rhythmic behavior, feeding and fasting, activity and rest cycle is important, because all our body hormones, enzymes, genes are all timed to be switched on and off based on the light and dark cycle. So when we mess around with this, then it leads to metabolic derangement. So this slide shows you how these clock genes, so we have a very specific network of these clock genes which regulate the expression of various other factors in our body, which affect the metabolic functioning of a cell during a daytime and the nighttime. So for example, in humans, there is a specific pattern of when this hormone, when a specific hormone will be released in our body. So the catabolic hormones are generally released during the daytime, because that time we are eating, so we need to digest it. At the night time, it is more of an energy conservation phase, because you won't be taking, and at the morning time you will be needing that energy. So if you start eating in the night, you will reset this all program and you will start storing fat, because that is the mode in which the night time is meant to be. And same thing goes for signaling and for transcription factors, which are part of the clock genes. In nocturnal animals like mice and rat, it is just the opposite, because they eat mostly in dark, so their catabolic hormones are all activated during dark phases, and in the morning, during the daytime, they are at rest. Okay, so this is the process of circadian rhythm. Now there were some studies done to see what if we time our eating to this circadian rhythm. So we have to synchronize our eating behavior to the circadian rhythm of the body. So these the scientists took these obese mice. So they were given high fat, they became obese, and then once they achieve a obese phenotype, they were again given this McDonald's burger. Okay, so I mean to say the high fat diet, and then they were given in two groups. One group was given ad libitum, so they were in the cage, this high fat was kept, and anytime they want to eat, they could eat it. But the other group is a set of obese mice which was given this high fat diet only during the night time, the time when it is supposed to eat. So you have to remember that unlike humans, their active phase is night and our active phase is daytime. Okay, so for them to be metabolically active, they are given at the night time, and in the morning the food is taken away. So they can only eat, they can eat as much as they want, but they can only eat during this time window of night and not during the day. So what they found is that when a mice is has been given this high fat diet all over the time during day and night, they eat and they become morbidly obese. But a obese mice which is given high fat food only in a restricted time manner. So this is not calorie restriction. So we all know about calorie restriction as a way to reduce fat. This is not calorie restriction. This is you can eat as much as you want. Calories are not limiting, but your time is limiting. So you have to finish everything between that time. So in a time restricted manner, given the equal calories, these mice are still obese, but they are fit. The meaning they are fit means their muscle mass is intact, their sugar levels are fine. They may still look obese, but they are metabolically active otherwise. Now what happens if you give a lean mice the same thing? So this is a poor lean mice, and he has been offered to eat high fat diet, but given two options: either eat it all the day, so the food will always be with him, or he can only eat during the night time when he's metabolically active, his enzymes and hormones are active. Okay, so what happens is that when a lean mice is given ad libitum high fat, he turns out to be obese. So he gains weight, he becomes obese. But when he eats the same amount of food in a time restricted manner, then he is fit, lean and fit. And what about humans? So this is a clinical trial done in human, inspired from these studies done in mice. So these scientists took human volunteers and gave them two options. First is to start their breakfast at 8 a.m. and then like the normal feeding habit we all have, and then last dinner at 8 p.m. And the other group of volunteers were asked to take breakfast at 8 a.m. but their lunch, so they won't have dinner in this case, but the last meal I'll say will be at 2 p.m. So this is a time restricted intake of calories, and calories are the same. They are giving the same food. They are not telling that don't eat carbo, don't eat fat. You can eat anything you want. but you have to finish eating by 2 p.m. And there are also studies which can extend it to 6 p.m. but not above that. And in both studies, what they found is that this time restricted feeding actually helps in reducing insulin, increases insulin sensitivity, beta cell function, appetite goes down, fasting triglycerides were up. So we don't know why they were up exactly, but other parameters were down. The oxidative stress was down, blood pressure was down. So it's definitely time restricted feeding is a lifestyle modification which has very positive effect on metabolic syndrome. And in some studies, although this study didn't show any effect on body weight, actually there was no change in body weight, but there are other studies which actually even show that they can lose weight. And even in this was done in pre-diabetic patients, pre-diabetic volunteers, and now studies are going on to see whether diabetics can also follow this regimen to control their blood glucose levels. So now we move on to the third part, exciting part of this research in metabolic syndrome. So most of you must have heard this phrase: "Let food be thy medicine, and medicine be thy food." And everything is in excess is opposed to nature, and all diseases begin in gut. This was said by Hippocrates, the father of medicine, in almost 400 BC. And now we are realizing that in fact he was right, because everything does start from the gut. So but the gut, not the human gut alone, but the microbes which live inside the gut. So studies have now shown that whatever we eat, so if we eat junk food, then our microbe, the intestinal flora is of a different type. And if we eat a lot of fibers and healthy food, the flora is of totally different type. And how food affect flora is an important area, that how some microbes flourish in a certain type of food intake, and other flora effect are existing in a different type of food intake. But whatever it may be, these flora, these microbiums in our gut secretes a lot of metabolites. So some of the key metabolites will be the short chain fatty acids. Is there basically they eat the things we have and they use it for energy, and these are the byproducts they make. They also affect the bile acids in our body, and which in turns affect the secretion from our gut of different hormones like the GLP-1, the hormone which I talked, regulating glucose levels. And this in turn affects our body glucose metabolism. So whatever we eat comes to us back through the interaction with our gut microbiome. So several diseases, even depression and other things, are now related to gut microbiome. So studies are now being carried out to manipulate these microbiomes, and how it could affect our metabolic well-being. So there are two strategies. First is the direct manipulation of microbiome, another is dietary manipulation microbiome. So when we eat a lot of junk food and a high fat food, then there is a lot of bad bacteria which start thriving in our intestine. So these are called pathobionts. So the aim is to use antibiotics, and these are all studies actually done and proven to be efficacious in animal models. So they have been already shown that in animal models these strategies actually work in improving metabolic well-being. So you can use antibiotic to kill off your pathobionts. It's only when you suspect your flora is mostly in the dysbiosis state. So the bad bacteria are more than the good bacteria. So you can kill off most of the bad bacteria. Then there's also a process of transfer of healthy microbiome. It's called fecal transfer. So this is actually done in humans in certain type of diseases like I forgot the name, but there are some intestinal problems in, I think it's okay, anyway. So there are some diseases in which actually fecal transfer has been done in patients, and it has shown that the fecal material containing healthy microbiome is transplanted in a diseased person, and that affects the intestinal well-being and general well-being of a person. Okay, so this is increasing the number of symbionts. This process, by these two processes, we increase the number of symbionts, so healthy good bacteria. The other way is by dietary manipulation of microbiome. So you give prebiotics or probiotics. So what is prebiotic? Prebiotic is anything you give which works, which act as a food for your good bacteria. Okay, so it may be a high fiber or other things, and probiotic is the good bacteria itself. So you can, you know, that those good bacteria capsules come and you can take them. So those are called the probiotics. And of course, by changing your diet pattern can also affect the percentage of your good bacteria versus bad bacteria in yourself. So all these strategies regulate your gut microbiome and gut interaction, and helps in the metabolic well-being. So lastly, to conclude, from these three major groundbreaking discoveries is that activating brown beige fat improves metabolic health. Chrononutrition may help to counter several aspects of energy metabolism in human, and manipulating gut microbiome using probiotic and fecal microbiome transplant may lead to positive result in improving glycemic control and metabolic syndrome. So with this, the take home message is eat good, eat on time, and burn it fast.